we need to talk about the production and development of an ovum, which is known as oogenesis. Talk about oogenesis, we're talking about mitosis of what's known as primordial germ cells within female embryos. Process produces diploid oogonia, which is basically telling you they're going to be these small cells which have 23 pairs of chromosomes. DNA is duplicated and they will give rise to what are known as the primary oocytes. These guys will have 23 pairs of duplicated chromosomes. Starting at puberty, once a month, every 4 weeks or so, depending on what the female's menstrual cycle is, follicle stimulating hormone, FSH, stimulates a single primary oocyte to undergo meiosis 1. That cell will give rise to what's known as a secondary oocyte, as well as polar bodies, which are these going to be these small, non-viable, lack of a better term, cells, because unequal cytokinesis will occur. The end products of meiosis 1 are primary oocyte, which is developed into the secondary oocyte, and the first of the polar bodies, one of those guys. And this shows you what's happening. So take a look at that and look through those. And then ask questions if you have them. You need to know what's printed in the power site, not necessarily every single detail about what's happening on this chart. Okay, so know the verbiage. You don't need to identify each step on this chart. Not asked that on the test. Okay, during childbearing years, we just went over this. Every 4 weeks or so, FSH is going to stimulate the maturation of one primordial follicle. Over a 14-day span, this is what happens. That primary oocyte will enlarge and will undergo meiosis 1. Follicular cells are going to multiply, giving rise to stratified squamous epithelium. These are going to be composed of granulosa cells. A layer which is known as the zona pellucida will form, which is going to separate the oocyte from those granulosa cells. That follicle is now called a primary follicle. What's known as the antrum will develop, and that is just a fluid-filled cavity. You have a crown of these granulosa cells surrounding the oocyte. That is known as the corona radiata, and the follicle is now called a secondary follicle. If that secondary oocyte is penetrated by sperm, meiosis 2 will kick in. You talk about meiosis 2, you're talking about equational cell division. When, and actually if, meiosis 2 gets completed, you have a second polar body that is going to be separated off from a large ovum, which contains 23 chromosomes. Sperm is haploid, and the ovum is haploid, and that create a diploid structure containing 23 chromosomes. So just as a point of reference, and I wouldn't ask this in terms of specificity, how big is a sperm cell versus an ovum? But this is kind of interesting to know. A red blood cell is about 8 microns in diameter. Sperm cell is considered, or is the smallest cell in the body. Male sperm is the smallest cell in the body. It's also the only human cell to have flagella. Male sperms are about 5 by 3 microns in size. In comparison, the ovum is going to be ginormous because it is around 120 microns in diameter. So way bigger than any sperm, way bigger than a red blood cell. It's kind of cool. That's a super good video on ovulation. We know that oogenesis, which is involving meiosis one, gets completed as the follicle matures. It takes around 2 weeks. On maturation, luteinizing hormone, LH, will cause the follicle to burst. Secondary oocyte will be released. And after ovulation, that secondary oocyte gets pulled or drawn into the uterine tube or the fallopian tube. Remember, we have cilia to help with that, to facilitate that, and to facilitate the movement of the oocyte along the uterine tube. This shows you what is happening in detail. And no, I will not put an image up on your test and say, okay, what's happening in this specific micrograph? But you have to look at this because this is spectacular. Look at that picture, not just spectacular. It's just amazing. Now, once the sperm cell has penetrated the secondary oocyte, we know that meiosis II occurs. It forms what's known as a female pronucleus, which is haploid. We mentioned that, 23 chromosomes. The sperm will drop or shed its tail, which is going to form a male pronucleus, also haploid, 23 chromosomes. They will fuse, forming what's known as a segmentation nucleus, containing 23 pairs of chromosomes. So let's compare oogenesis versus spermatogenesis. During a lifetime, oogenesis begins in the fetus and ends at menopause. For males, spermatogenesis begins at puberty and continues on and on and on and on and on and on and on to death. Not talking about how viable the sperm are, I'm saying it continues. In terms of the number of functional different gametes, there will be some major differences. Oogenesis produces a single viable haploid ovum with between 2 or 3 polar bodies, which are also haploid. Spermatogenesis is going to produce 4 sperm, all of, we hope, whom are viable. Error rates will also differ. Oogenesis has an error rate of about 20%. Spermatogenesis is relatively error-free, relatively speaking, because it has an error rate of 3 or 4%. That shows you a lovely chart which came out of the book about spermatogenesis versus oogenesis. In the course of a lifetime, males produce about 500 billion sperm. That's pretty moving. Talk about the ovarian cycle in a minute.